Okay, so welcome back, students. Am I audible to all?
Yes, sir.
Okay, so किसका presentation कटाना आज?
Athoiba and me, Yukta.
Okay, so who is giving first? So Yukta, you are presenting first or Atoiba? 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 Atoiba didn't join yet. Okay, so Yukta, you can start. Meanwhile, Atoiba will join.
Okay, sir. Sir, are the slides visible? Hello.
हाँ युक्ता दिख रहे हैं, start करो.
Okay, so
And
good afternoon, everyone.
turn on your camera also.
Sir, I have not joined from my laptop.
हाँ तो कोई बात नहीं, मोबाइल
broken.
तो मोबाइल से भी कैमरा ऑन हो जाता है ना.
Okay, sir, फिर आवाज नहीं आएगी. एक मिनट. Am I visible now? Okay, so good afternoon, everyone. My topic for today is chromosomal and mitochondrial DNA. So, moving to the introduction slide. So first, I'm gonna give a brief introduction about what is DNA and who discovered it, as we have already studied, as Nandini discussed it yesterday. So I'm gonna give just a few pointers. So DNA was discovered first by Friedrich Michel in 1869. DNA is found in all prokaryotic and eukaryotic cells and in many viruses. It codes for genetic information for transmitting the inherited traits. And the double helix model of DNA was given by James Watson and Francis Crick in 1953. And a contribution in the same field were also made by uh, Albert Albrecht Kossel, Avery, and Erwin Chagaff. DNA is found mainly in nucleus and in mitochondria of the cell, and also it is not present freely in the cell. Instead, it is well packaged and highly ordered. So DNA molecules bind with the protein structures called as to form a structure called as chromosome. However, in the mitochondrial DNA, it is present in the circular form, and it was discovered by Margaret M.K. Nash and Sylvain Nash in 1960s. Moving to the next slide. So, a double helical structure of DNA. It was, as I mentioned, was given by Watson and Crick. And DNA is a double helix polymer that consists of two DNA strands. It looks like a twisted ladder, as you can see here. So it is made up of nucleotide monomer units, and the nucleotide consists of sorry, nucleotide consists of a deoxyribose sugar molecule to which the phosphate group and one nitrogenous base is attached. The nucleotides are joined together by the covalent bonds between phosphate of the one nucleotide and sugar of the next, forming a phosphate sugar backbone. So one turn of helix contains 10 base pairs, and each base pair is separated here by 0.34 nanometer, or we can say 3.4 angstrom. So it is a negatively charged molecule, we all know, due to the negatively charged phosphate ions in the sugar phosphate backbone. And the N base can only pair in a certain way. Adenine pairs with thymine and cytosine pairs with guanine. The two strands are attached by hydrogen bonds between the bases, and both strands are complementary to each other. They run opposite to each other and are called as anti-parallel strands. And the DNA replication is also the process that occurs in all living beings as the most essential part for biological inheritance. So it was just a brief knowledge about the helical structure. Now moving to the main topic, chromosomal DNA. So the nuclear DNA or the chromosomal DNA is inherited from both the parents. Each cell contains only 23 pair of chromosomal DNA. The nuclear DNA is organized into linear strands that make up to the 23 pairs of the chromosome in human genome. And there are about 3.2 billion base pairs in the nuclear DNA of the cell. Buried in these linear strands are about 20,000 genes that code for proteins, along with other sections that control genes that are expressed in each cell. The chromosomes that comprise the nuclear DNA are paired, and each member of a chromosome pair carries the same gene and codes for the same trait. Differences in the nucleotide sequence of the gene are responsible for the variety of traits in the biological organisms that we see. Moving to the next slide. Packaging of chromosomal DNA, how it is packed such a long length. Like we know, if the entire DNA of human body is stretched, it would be approximately 1.2 into 10 to the power 10 miles, which is about the 6 rounds of Earth. So DNA packaging facilitates the accommodation of this large amount of DNA to fit in a cell, which is very small in size. So the process of packaging is complex in eukaryotes than prokaryotes. And histone is the protein that allow the DNA to be wrapped tightly and packed around it. So the structure in which the negatively charged DNA is wrapped around the positively charged histone is called as a nucleosome, and each nucleosome is formed of DNA wrapped 1.6 times around around 8 histone protein molecules. So it is further condensed and supercoiled into structure that we call as chromosome. Moving to the next slide. So here is the diagrammatic representation of what I just said, how the chromosomal DNA is packed so that it could fit inside the nucleus. And the main role here, as I mentioned, are of histone structures. The histones are very highly basic proteins. They're rich in lysine and arginine. And the main protein component, they are the main protein component of chromatin. So until now, there are 8 types of histone protein that have been discovered, but proteins that are associated with this packaging of the chromosomal DNA is H1, H2, H3, and H4 histone protein. This protein make the dimeric units, forming an octamer together, which is globular in shape. And DNA is then wrapped around it, forming a nucleosome unit. And the various nucleosome units form a chromatin, and then we see these chromatin forms these loops, and which are further condensed and form ultimately the chromosome. And in order to make the DNA active, if we want the DNA to be active or worked in various processes, it is unwrapped, and the histone proteins detach themselves from the DNA to make it accessible for other classes of protein. So moving on to the next slide. So another important class of protein that I like to briefly mention here are histone chaperones. So histone chaperone, the association of histones with these chaperones complex is important to maintain the genome and epigenomic integrity. The histone chaperones are proteins that regulate the pathway of assembly and disassembly of nucleosome. They help in the histone transport from cytosol to nucleus via important histone interaction, and some act also as a reservoir of the histone. Example is NAS, and they also regulate the histone modifying enzymatic activities and are involved in DNA binding and assisting in nucleosome assembly. Moving to the next slide. So the next topic is the mitochondrial DNA. All the mitochondria for an individual is provided by the egg cell, meaning that all the mitochondrial DNA or empty DNA is inherited only from from the mother or the maternal line. So the nuclear DNA resides only in the membrane-bound cell nucleus, whereas the mitochondrial DNA molecules are found in the mitochondria, which are scattered throughout the cell cytoplasm. So here is the representation of human mitochondrial DNA, showing the arrangement of regions that code for protein and the RNA molecules. So the mitochondrial DNA is circular, and it contains about 16,000 base pairs, and there are only 37 genes and a non-coding region. The non-coding region is called D loop, that does not code for any gene product. And of these 37 genes, the 22 codes for tRNA, 2 codes for rRNA, ribosomal RNA, and 13 codes for the protein that are important for the cellular energy production. So a cell may have 10 to hundreds of mitochondria, each with hundreds of DNA molecules. For example, in human liver cells, they contain more than thousands of mitochondria with hundreds of circular DNA molecules inside them. So the mitochondria also, we say that the paired chromosomes are diploid, though we see that the mitochondrial DNA have multiple copies of their circular DNA molecules, and they are not paired. So we can say that they are haploid in nature. So when nuclear chromosomes are replicated during the process of creating new cells or crossover or the recombination between the members of the chromosome pairs, it ensures variety and uniqueness to the new organism. So no recombination we see in the mitochondrial DNA because there is no pairing. Moving on, moving on to the next slide. The unique features of mitochondrial DNA. So first sir, is the 9th slide visible to you? It's not visible to me.
It is not visible to me also. Your still mitochondrial DNA is there. Unique features of mitochondria.
So just a second, there is some network issue. Sir, is it visible now?
Yes, it is.
Hello.
It is visible now. You can continue.
Hello.
Yes, Yukta, continue.
Okay, sir. So the unique feature of mitochondrial DNA that I want to mention is high mutation rate, the unique genetic code, and heteroplasmy. So what are these? In high mutation rate is that the mitochondrial DNA lacks the repair mechanism and the proofreading capabilities that ensure the integrity of the nuclear DNA during the replication. So this leads to the mutation rate for mitochondria to increase 10 times higher than that of the nuclear DNA. So unlike the nuclear DNA, the mitochondrial DNA is densely packed with functional genes with no duplicates for backup in case no gene is disrupted by the mutation, and the mutation in these coding areas results in mitochondria deficient in gene products, mostly which are proteins that are necessary for their function. So they will most likely be unable to reproduce and pass to their mutated genes. And despite the higher mutation rate, the functional part of the mitochondrial genome or mtDNA varies very slowly among the individuals over time. So the mutations in the non-coding region of mtDNA do not harm to the mitochondria, or nor do they affect the survival of the organism. So they accumulate and then pass down through the maternal line. So these mutation also provide a source of variability that is useful in human identity testing and investigating the evolutionary and genealogical relationship. Second is the unique genetic code. So the mitochondrial genetic code differs some from the code of the nuclear DNA or the chromosomal DNA. So, for example, in the mitochondria, the codon AUA codes for the amino acid methionine, whereas the same codon in the nuclear DNA codes for isoleucine. So similarly, UGA codon codes for tryptophan and is not a stop codon as it is in the nuclear DNA or the chromosomal DNA. Also, the nuclear DNA has long stretches of non-coding DNA throughout the genome, and these non-functional nuclear DNA segments called as introns make up as much as the 94% of the nuclear DNA, and mitochondrial DNA has no introns. So it is found that that only 55 base pairs of the mitochondrial DNA have no known functions. The third unique feature is heteroplasmy, which is basically the presence of the normal and the mutant DNA in different proportion, which are present in different cells. And it is also considered important for determining the severity of any mitochondrial disease. Moving to the next slide. So this last slide basically highlights the key differences between the nuclear chromosomal DNA and the mitochondrial DNA. So like I mentioned, the nuclear DNA is inherited from both the parents. The mitochondrial is inherited from mother only. The nuclear DNA is linear. The mitochondrial is circular. The nuclear DNA size is far more than the mitochondrial. Like it's 3.2 billion base pairs here, and for mitochondrial, it's around 16,000 base pairs. And the number of genes also varies. There are 23 pairs of nuclear DNA in each cell, and there are thousands of mitochondrial DNA in each cell, 100 to thousands. And the nuclear DNA is paired or diploid, and mitochondrial is not paired or haploid. They are also varied by recombination. Proofreading mechanism is not present in mitochondrial DNA, and the mutation rate, as I mentioned, is high, very high in the mitochondrial DNA. There is unique genetic code in mitochondrial DNA. Mitochondrial DNA do not have introns. And the last point, that the nuclear DNA is monocystronic and the mitochondrial DNA is polycystronic. So what it means that the nuclear DNA produce mRNA that codes for only one protein. So if there are more than one polypeptide chain coded for in the nuclear DNA nucleotides, the resulting mRNA is cut apart into separate mRNAs before binding to a ribosome. So the nuclear DNA is monocystronic, meaning that there is a separate strand of mRNA for each protein, and mitochondrial DNA uh, I'm sorry. Mitochondrial DNA has a scheme more like that of the eukaryotic cells responsible for its origin. So, if an uninterrupted segment of mitochondrial DNA that consecutively codes for, let's say, 3 or 4 proteins, uh, and is transcribed to mRNA, then that mRNA is not cut into 3 different mRNAs, then the whole mRNA proceed to bind to a ribosome, which forms the which will code for all the 3 proteins or the 4 proteins. So, so in contrast to the nuclear DNA or chromosomal DNA, the mitochondrial DNA is polycystronic, meaning that on one strand of mRNA can be responsible for producing more than one proteins or encoding more than one gene. Thank you. This was the end of my presentation.
Very nice presentation, Yukta. And I hope all of you got quite good information about the
Sir, your
chromosomal
voice
DNA.
is very low, very low.
Just a moment.
Okay, is it better now?
Yes, sir.
Okay, so very nice presentation, Yukta, and Yukta covered most of the points, and I think she covered more than what I expected, and even the thing about the DNA she covered, said about the chromosomes. That that was a part of Atoiba, but Yukta herself presented that. Okay, so one
Sorry, sir, I
thing
didn't know
you
that.
could include that point that will help you in making others to understand better way. One thing is that if you move to your slide number 9, you mentioned that the high mutation rate and second unique genetic code. So this unique genetic code you should need to explain a bit more. And this unique genetic code, they are not means when we are saying uniqueness is a bit less as compared to when we are saying about the chromosomal DNA. The genetic code, that code for amino acid in your chromosomal DNA may code for another amino acid in case of your
Yes, sir. Yes, sir.
chromosomal
It codes for
mitochondria.
answer.
So that we refer the degeneracy of genetic code. So genetic codes are not degenerate in your mitochondria. Okay, so this is a thing that you should know.
Okay, sir.
And while explaining it, if you show some of the codes, like if you say that this code if is present in your chromosomal DNA, then it is going to code for X amino acid. In case of mitochondrial DNA, it is going to code for Y amino acid. So if you show those examples, then it's going to be a bit better explanation. And secondly, thing that you were explaining about the monocystonic and the polycystonic, you explained well, but the thing is that we were not having any picture in front of us that will help us in the understanding. But you,
Yes,
I
sir. Yes, sir.
tried to explain a bit well. And as she mentioned, the mono stand for one, and cystone means one mRNA, as you mentioned, and one protein meant one that mRNA is going to unique. What's from the start codon to the end codon, the stop codon that she was saying, there is only going to be only one protein. That means that mRNA is going to code for one protein. But in case of the polycystonic one, mRNA is still there, but it is going to have different frame, different start codons are going to be there within your mRNA. So with the different location, your protein synthesis can start. So a single mRNA may code for more than one protein. And this polycystonic is a property of your viruses also. So viral genome is mainly polycystonic. And that one more thing, the overlapped genes, one term is there, means genes are overlapped within, they are not distinct. Within a same sequence is going to be part of one gene, and it's also going to be part of the second gene also. This is a thing that I think I should explain a bit more. Rest, I think Yukta have explained very well. And now presentation is open for discussion. So who will, I think lots of you are going to have some queries from Yukta. Who will ask question from Yukta? So सभी चुप है, कोई बोल ही नहीं रहा हो या चले गए सभी, सो रहे हो? Are you sleeping or still?
No, sir.
युक्ता सब सो गए, देखो तुमने कैसा प्रेजेंट किया, सिर्फ तुम्हारी सहेली नम्रता जगी हुई है, बाकी सब
present.
अच्छा मनस्विनी भी जगी हुई है. Yes, Demetrius.
Okay.
Demetrius want to ask something, Yukta. बोलो भाई.
Yes, sir. Am I audible? Yes. So my question was to Yukta ki,
Yes,
you know,
Demetrius, ask.
what, sir? Yeah, so my question was that mitochondrial DNA can be like harmful sometimes if it mutates. So how will you prevent when it's being transferred from a mother to their offspring?
Okay, so like I mentioned that the mitochondrial DNA is transferred or inherited through the maternal line only. So if we want to prevent it from inheriting or transferring to the progeny, what we could do is that we could prevent the transfer of mitochondrial DNA from the mother by introducing mitochondrial DNA from, let's say, a donor female. So, in uh, like techniques like in vitro fertilization, we could do that.
Demetrius, the issue is that you can't prevent mitochondrial DNA to transfer. Because if you
So,
prevent mitochondrial,
if we are not using the he said, if we want it is mutated, the mitochondrial DNA in the mother is mutated, it's harmful if it transferred to the progeny. So we could use a donor female, right?
prevent to the transfer, then child will die. Then you are saying the donor, and donor is going to be mitochondrial donor, whole egg is mutate. And it's going to be completely gene therapy type of thing that you take out the egg from the mother and in that micro injection you need to insert a new mitochondria there. So microinjection type, I don't think it is a feasible thing, but mitochondria is the essential part. Know it. But gene therapy you can utilize for. Okay, so any other questions? Except Sam, no one asking. Manashwini? Manashwini?
Yes, sir.
No query from your side also?
No, sir. She explained it very well, so I don't have any questions.
When in exam, you do not answer the question. Okay, it's fine. So next is going to be Atoiba, and Atoiba is absent today. So he is going to be marked 0. So for you people, those who have presented, I have created an assignment. All of you, I think, can see the assignment in your assignment box. So as you're done with your presentation, you have to upload your presentation in your assignment box. Okay. Like Nandini and Yukta and who was the fourth? Saiba. Saiba, Nandini, Yukta, all of you upload your presentation in the assignment box. So it is a 10 marks presentation. So just by presenting and giving a good presentation, you can assure 10 marks. Okay.
Okay, sir.
Okay, sir. I'm
Okay, so now as the Atoiba is missing today, so but we still have a few more minutes. I think around 20 minutes we have. So during 20 minutes we can discuss something, and that Atoiba was have to present. So for that I would like to take the help of Yukta's slides. Yukta, please share your slides again. Yukta.
okay, sir.
Okay, so I'm taking control of it. The slides are moving.
Yes, sir.
So, yeah, so she explained you about the double helical structure, and this is a thing that I want to mention. So the term, if anybody asks you what you mean by the chromosome, so that most of the time we got confused what this chromosome really mean, because generally we know that DNA, and is the DNA and chromosome both are same thing or they are a bit different. So and generally what happens, we use DNA and chromosome just as a synonym of each other, but they are not. When it is being asked that the genetic material, we said that the DNA is genetic material, but we do not say that the chromosome is genetic material. But the chromosome is one which is carrying the genetic material. So then it means chromosome is a bit different from your DNA. So the difference is there in its definition itself, simply when we are going to define it. So the chromosomal is nucleoproteo complex. Simple thing. Okay. So two terms I used here. One is the nucleo. Nucleo stands for the nucleic acid. It means it is the nucleic acid is there. That is your DNA. And secondly, the proteo. That means the proteins are going to be there. So nucleic acid and proteins are there. And then the term is used term complex. That is a complex of DNA and protein. So DNA and protein complex is called chromosome. And whenever all of you are seeing in this image some X type of structure, so whenever your DNA is there in such X type of structure, then only we are going to use term chromosome. Okay. So the term chromosome that we are using chromosomes mainly for this X1. And just a moment. Okay, so this X structure we are going to call it a chromosome. And again, this X type of structure we are going to be able to see only during the cellular division. That is going to have X. It is going to have two chromatids. One chromatid is going to be, both are to them we call the sister chromatids. And each chromatids, these two chromatids are going to be there only in the cellular division when DNA replicating. If DNA is not replicating, and then you are not going to have this X type of structure. This X type of structure is going to form only after the DNA replication, because each of these chromatids contain double-stranded DNA. So simply you can see that in this X type of structure, your DNA is tetrad. These 4 strands are there. So DNA is not double-stranded in this chromosome. In the chromosome, your DNA is going to have 4 strands, and these 4 strands are present where? These 4 strands are present at the centromere, where your DNA, where the both chromatids are joined together. Okay, so then again we move to the chromosomes. When we are talking the chromosomes, then before replication, what it is going to be? So when replication is not there, at that time we are using term chromatids or chromatin for the chromosomes. Okay, however, you can also call them chromosomes, but we are using term chromatin. So chromatin is a single chromatid. So one chromatid is there. It means your chromatin is going to have a double-stranded DNA, and your chromatin is going to, again, chromatin is also a nucleoprotein complex. So there also nucleic acid is there, protein is there, and the complex is a chromosome again. Okay, so the chromatin is a double-stranded DNA. Only a single set of DNA is there in your chromatids, not as in case of chromosomes. A double set, 2 set is there, tetrad, or a 4 strands type of things are there. So each chromatid have 2 strands, and at the joining or at the point of your centromere, there are 4 strands. So tetrad are there, means 4 strands are there in there. Okay, and that of somewhere of you, if I think Nandini, when she was describing DNA, and at that time she mentioned that 4 stranded DNA also. Nandini, you mentioned that during that time? Double-stranded DNA, 4-stranded DNA.
I'm sorry, sir. Your voice, I can't hear it.
All of you are not able to hear my voice?
So we can hear.
So it's very shaky on my end, probably because there was rains and they're still fixing the Wi-Fi. So the problem is probably on my
Is
side.
it better now? Is it better now?
Yes, sir. It's
Okay.
better now. Thank you.
So this is a 4-stranded structure that is there in at your centromere. So this is another type of DNA. When Nandini was explaining, we always used to use single-stranded DNA, double-stranded DNA, and the 4-stranded DNA is also there. And the 4-stranded DNA is there in chromosomes when the at the location of the centromere. Okay. And now within this chromosomes, as if you are able to zoom its structure, you can see that different chromosomes have different size and different location of your centromere is there. So in this X-shaped centromere, both the side of the centromere are not equal. There is asymmetry. And this asymmetry is there because of the position of your centromere. So on the basis of the position of centromere, also we can classify your chromosomes, that where the location is. So if it is at the center, called the metacentric. If it is a tip, we're going to call it acrocentric. At the tip, we call it the telocentric. Just below the tip, we call it the acrocentric. So depending on the location of your centromere, we also can classify your types of your chromosomes. Okay. So then how to distinguish the arms? Like as I'm saying you that there is a dissymmetry in the position of your centromere. So possibilities are there. One side of the arm is going to be bigger and other is going to be smaller. Okay, so now this on the basis of the smaller and the larger arm, we name it. So which arm is larger, which arm is smaller. So that is the first I'm telling which is going to be smaller one. So to smaller arm, one term is used that called petite. So I think genetics, you might have heard the term petite. So petite means small. So smaller arm is a petite arm. So also we are calling it as a P arm. P stand for petite, and the smaller arm we are describing P arm. Okay. So the now what should we give the name to the larger arm? So larger arm just randomly as to the smaller arm we are calling P arm. So larger arm we are calling Q arm. P Q R S like it is just P. Next to the P is Q. So Q doesn't means the larger. Okay. So P is the petite smaller one, but Q is a larger one because after P it came the Q. Okay, just it's for the larger arm there is no clear definition that why we call Q to the larger arm. Okay, to the smaller arm we are calling a P, and to the larger we are calling Q. So these P and Q arms are there because of the location of the centromere, which give the asymmetry in the sizes of these arms. Okay, so this is again a bit few things about your DNA, about the chromosomes. And when we are referring the chromosomes, as I mentioned you that we are describing your chromosomes as a nucleoprotein complex. So how this complex is forming, as Yukta mentioned you that this complex is formed around proteins, and the proteins that you are going to use to make this complex is the name of the protein, histone. So histone is a protein that you utilize for the making this complex, and but all histones are not alike. So there are different types of histone. How many histones are going to be there? Only one histone, two histone, three histone, four histone. So there are totally 5 different types of histones are there. So in your chromosomes, there are 2 types of protein. One is going to be histone in nature. Second is going to be non-histone. So 2 types of proteins are there in your chromosomal packing and your chromosome. One is the histone protein, and second is the non-histone proteins. So here when we are describing your chromosome, we mainly talk about the histone protein because they are the major protein. Other than these, also some other proteins are also there, but they are not the major component. So that's why we are not referring them much frequently. Okay. So as I mentioned, the histone is the major protein in your chromosomal packing, and there are 5 different types of histones are there. So 5 different types means H1, H2, H3, H4. Okay, and then from where the 5 is there? So H5 is not there. So 5 type, but H2, H3, and H4. So 5 type came from the H2 itself. So H2 is another of 2 type, H2A and H2B. So total become the 5. H1, H2A, H2B, H3, and H4. So no H5 is there, but total proteins are 5. Okay, so just checking any new slides is there in which. No, she's not describing that one. So, okay, so these 5 proteins are there. So in these 5 proteins, further 4 proteins have different roles, and 1 protein have a different role in this packaging. So 4 protein that I'm mentioning, I'm just calling that H2A, H2B, H3, and H4. So these 4 proteins are there. Now 2 unit of each. So total proteins are going to become 4 into 2, 8. So these 8 proteins, they join together and they form. Okay, so I think I will share you my slides. So okay, so we'll do that in our next class. I think our time is also rushing out. So just do. Okay, so these 8 proteins, they join together and they form 8. So that's why we call this term octamer. Means that unit that have 8 subunits. Okay, so the histone octamar is going to be formed. Histone octamar is going to form with the use of 4 different type of histones, and each is going to be 2 in number. If the H2 protein, H2A, 2 unit of H2A, H2B, 2 unit of H2B, H3, 2 unit of H3, H4, 2 unit of H4. So these all they come together and they form a complex, and that complex we call the quaternary proteins. So quaternary complex, tertiary is individual one. If two proteins join together and form a complex, it's called the quaternary one. So this complex of 8 proteins forms, and it we call the octamers. So DNA is going to wrap around this octamer. So this octamer, when your DNA is wrapping, so when the DNA is wrapping, then it turns around it. So how much turn it forms? 1 turn, 2 turn, 2.5 turn. So as when you're talking about the DNA, it's helix, 1 helix, 2 helix, double helix. So how much turn it is, how much time it is wrapping around it. So it is going to wrap. It is not wrap completely 2 times, less than 2 times it wrapped up. I think 1.8 times it wraps around your octamer. So then this octamer is going to be when DNA goes in and goes out. So 2 arms are going to be there. Here you can see in it one spindle shaped protein or a cylindrical type. What is this? You say egg shaped protein is there. That is joining these 2 arms. So that is going to be H1. To that we are calling histone 1. Okay. So that H1 protein is going to be there, and that is playing a role of linker. And it is what it's doing, it is linking. Whom it is linking? It is linking to a nucleosome. So this wrapping of DNA around your histone, we are calling it a nucleosome. This is a unit. This is a first packaging unit. Single packaging is there. First level of the packaging is there. To this first level of packaging, we are using term nucleosome. So nucleosome is one DNA wrapped around octamer histone and linked with the linker histone is called nucleosome. Okay, so this is just a simple description of your nucleosome. Okay, then the level of packaging also going to be higher. This nucleosome is going to be further packed up, as you can see here in this slide, as Yukta shown you. So this is each individual nucleosome being wrapped around that second, third, fourth, fifth. So these 4 nucleosomes are there. Now these nucleosomes further wrapped around each other. That will give you a second level of packing, then third level, and then the fourth level of the packing are going to be there. So that in end, this packaging leads to the formation of your chromosome. Okay, so this is just a glimpse of your chromosomes, and I hope all of you have read these things. Just I revised you again. Okay, so today Atoiva missed the presentation, so it means he missed the marks also. So this is the things, no one is going to get a second chance for the presentation or for the exam. Okay, so be stick with the time and be vigilant also. So don't give excuses that sir I forgot or I did this thing, I was not well. If you are if anything is critical is happening with you, then you need to inform me in advance that sir this is an issue and I can't give the presentation because of this issue. Okay, so now as we are ending our session, so please turn on your camera while I'll call your name for recording attendance. So first is Adish. Adish, you can unshare the slide. Now you can unshare the slide.
Present, sir.
Adish, also turn on your camera. Ayushi.
Present, sir.
Demetrius. Demetrius.
Present, sir.
Devanshi. Demetrius, turn on your camera. Ishita.
Present, sir.
Janvi.
Present, sir.
Kshitij.
Present, sir.
Manaswini.
Present, sir.
Nandini.
Present, sir.
Namrita. Nehal.
Present, sir.
Rupal.
Present, sir.
Sahiba.
Present, sir.
Anjana.
Present, sir.
Shakti.
Present, sir.
Shivani.
Present, sir.
Tanvi.
Present, sir.
Tridev.
Present, sir.
Vivek.
Present, sir.
Yukta
Present,
and
sir.
Yuvraj.
Present, sir.
Okay, so Yuvraj and Vivek, turn on your camera.
Yes, sir.
Okay, so thank you all. Stay safe, stay happy. See you again next week. Till then, enjoy the week.
Thank you, sir.
Thank you, sir.
Welcome.